a postdoc at the USDA, working under Dwayne Shoemaker. I'm new to the world of ants. I'm actually a butterfly guy for my PhD, but it's, as you probably already know, quite an amazing system to work in. So I'll give you a little bit of background about social parasitism or ant social parasitism more specifically, in case you're not aware. Social parasitism is basically, or ant social parasitism is when two ants share the same nest. Either one of them exploits the other species. And it's evolved independently multiple times in Hymenoptera, in ants specifically. Of the over 12,000 described species, there are 230 known social parasites. This is probably an underestimate because social parasites are usually relatively rare. There are probably more than that. And there are four types of social parasitism. One is this xenobiosis, or guests. I like to think of it as the Airbnb social parasitism. They kind of just hang out in the nest. They have their own worker caste. They take care of themselves, but they're using the nest as a shelter. Then there's the temporary parasitism. This is like a Kickstarter parasitism. They come in, they kill the queen, they hijack the colony, and then they use it to get themselves started and build their own colony and then replace the colony that they invaded. There's also the doulosis, or the slave-making ants. So they'll infest a colony. They don't have a worker caste. They kill the queen. They basically hijack the host, the workers of the host, and use that to raid other colonies and create new slaves. And then the most common social parasite is an inquiline social parasite. These have no worker caste. They invade a colony and produce just reproductive, so queens and males, and use their host, keeping the queen alive, to produce basically food for their offspring. So the species I'm going to be talking today is Solenopsis dageri. It's native to South America, and it's an inquiline, so an obligate social parasite. It requires its host to live. Like many social parasites, it's evolved morphological adaptations, physiological adaptations to suit its lifestyle. So here they have much less serrated mandibles, about big that won't chop the head off of their host queen. They also have some way of chemically camouflaging to get in undetected into their host colony. And then they're also much smaller, this is typical social parasites, than their host. If you look in the field, they're, like I said, they're quite rare. So 1 to 24% of the colonies are actually parasitized where they're found. And on average, it's only actually about 5% of the nests that you'll find out of the field have a social parasite infestation. But when they do infest the nest, they can be quite damaging. So they'll actually dramatically reduce the production of the host's workers and reproductives, and eventually this can lead to the collapse of the host colony. So for dageri, there are 5 known host species other than Kevin Spacey. These are in the Civissima complex, so these are fire ants. They're native to South America, and 2 of these species have been introduced to the United States. So there's Invicta and Ristra. This is the imported red and imported black fire ant. And so a while ago, 70 years almost now, dageri, there was a study to look at potential biocontrols for stopping the spread of the fire ant. And dageri was an ideal candidate for use against the fire ant, hence the USDA's interest in dageri in my talk here today. But what is interesting is that most inquiline social parasites are usually highly host-specific, and they have a single species of hosts that they parasitize. So the finding that there's 5 different hosts for dageri seems it's very odd, it's very unusual. And it raises the question, is it really just one species of dageri, or is there actually multiple things in South America? And also an important question for biocontrol is there any host specificity? So if there are multiple things, are these multiple forms or species of dageri host specific? I should also say that for the hosts, so in fire ants, there's actually social polymorphism. So there's a supergene, speaking of supergenes, it's a large inversion. containing a large number of genes that basically, if you're heterozygous for big B, little b, the workers will not kill invading queens, and they can tolerate multiple queens, so this becomes a polygyne nest, where if they're homozygous for the big B allele, they will kill any intruding hosts, so they're a monogyne colony. So there's the social polymorphism, and that's relevant to social parasitism because it's thought that social parasitism may have arisen in a polygyne colony as a cheater queen, and due to the tolerance of multiple queens, it's thought that perhaps Daguerreae will have an easier time invading polygyne nests, and they also tend to be larger, which inquiline species tend to like larger colonies, species that produce larger colonies. So with that, the aim of this study was to basically characterize the genetic variation of Daguerreae within its host range, or native range, including the host, and answer three major questions. Are the genetically distinct forms within Daguerreae? Do we see any evidence of host specificity? And does the prevalence of Daguerreae correspond with the social form of its host? So this is the known locations of Daguerreae in South America. For this study, Dr. Shoemaker and Kenneth Ross at the University of Georgia went out and sampled 10,000 colonies. This is a lot, digging up fire ant nests. 9 sites, so 6 in Argentina and 3 in Brazil. If we look more closely at one of these sites in Argentina, so each site has about 40 nests that were sampled. In Brazil, unfortunately, that's more like 10 to 13. This is what it looks like out in the field, these nests. And for each nest, so here blue is the parasite, red is the host. Duane and Ken collected the DL8, so the wingless queen, and then the winged queen and males, and the worker. And then from a determine the social form based on a diagnostic allele that I told you about, and then constructed the phylogeny based on a mitochondrial data, partial colon gene sequence. So here's the microsatellite data for the parasite. And what you can see is there's 4 distinct clusters, and they're kind of broken into a hierarchy of this cluster being the most differentiated from these 3, greater amount of variation explained on the first principal components axis. These 3 are this cluster divergent from, or differentiated I should say, from these 2, and then these are the most closely related. These 2 clusters are found in Brazil, and these 2 in Argentina. And we'll come back to that in a second, but let's look at the phylogeny of the ants that we've collected here. So for these, now I've colored these by the mitochondrial haplotype and constructed a phylogenetic tree based on the Cohen sequence. And what you can see is that Daguerreae, this most differentiated cluster, is a monophyletic clay. These 3 form also a monophyletic clay, both sister to Electra or Pusilignus. And just for reference,